It's a privilege for me to introduce Dr. Dhandapany Perundurai. We call him Dan, and Dan is my postdoc mentor. I know him since 2016 when I joined his lab as a postdoctoral fellow. So I worked in inStem for almost 4 years and for a brief period in OHSU. And Dr. Dhandapany Perundurai is Scientist E and Assistant Investigator in Center for Cardiovascular Biology and Disease at Institute for Stem Cell Science and Regenerative Medicine, Bengaluru. He is also associated with Oregon Health and Science University, Portland, USA, as Assistant Professor of Medicine, Division of Cardiovascular Medicine, and School of Medicine. He has various prestigious awards, but out of those, two prestigious awards are Intermediate Indian Alliance Wellcome Trust Fellowship and American Heart Association grant. He is known for his famous work on MYBPC 25 base pair deletion and its association with cardiomyopathies in South Asian population. It is very well cited, highly cited work from him as a PhD fellow. He is Executive Council Member for International Academy of Cardiovascular Society. He is one among the best cardiovascular researcher I know. So because he is the one who started or initiated the stem cell work and variety of transgenic animals in India to explore various novel genes, drug repurposing for cardiac diseases. Over to Dan.
Yeah, thank you, Anupam. Thank you for the kind intro. And it's indeed very proud. Like Anupam is one of my students and who is now there as an assistant professor. And part of the work which I'm going to show now is where Anupam is also involved when he worked here as a postdoctoral fellowship. And Anupam was very instrumental in actually making a stem cell lab at my place. So thank you, Anupam, for that. And I will start sharing the screen. I think you're able to see the screen. Yeah, are you guys able to see the screen? Hello?
Yes, Dan, we can share.
Okay. So I belong to cardiovascular biology and disease team at Institute for Stem Cell Science and Regenerative Medicine. So for today's talk, I'm going to talk about briefly about how cardiogenomics and stem cells and precision medicine.
Actually, I can't see your slides, Dr. Dhan. We can't see it. On my computer, I can't see it. I don't know about others.
Oh.
Visible here.
It's showing that viewing Dan's screen, but I can't see your slides. They're all blank.
I see. Maybe I can redo it again.
Now it's coming.
Oh, it came. Oh, sorry. I just
show this. Yeah.
Yeah, I will again start it because I thought like there might be a glitch. Sorry for that.
Please all keep on writing your Q&A block, your questions in the chat box or the Q&A box, which will be taken in the last of the.
And now you are able to see it?
Not yet. I think it's trying to connect.
I see.
Yes, Dan, it's yeah.
Okay, let me know if I can proceed.
Yeah, now I can see it.
Okay, great.
Yes, thank you.
Thank you very much. So to start with, so the problem that I'm going to address today is hypertrophic cardiomyopathy. So hypertrophic cardiomyopathies are a group of heterogeneous heart muscle disease leading to thickening of the heart as shown here on the right side picture compared to a healthy heart. So here what happens is there will be an increase in septal thickness as pointed out here compared to the healthy heart, and ultimately that leads to diastolic and systolic dysfunctions. So 1 out of every 250 people are affected with this condition, and the causes are mutation in sarcomeric coding genes and signaling genes. However, 50% of the causes are still not known, and the South Asian populations are not promptly explored. First of all, why HCM gene discovery? The current HCM therapies, if you look into the current HCM therapies, they all rely on gene mutations. For example, for the myosin binding protein C3 or the myosin heavy chain, the two major sarcomeric gene mutations that causes the cardiomyopathies, the recommended therapy is basically a myocardial drug called mavacamten from the Bristol Myers Squibb. Whereas if the mutation is in LAMP2, then the recommended therapy is an AAV9 based therapy. In contrast, if it is DMD, then it is basically an antisense therapy. So for TTR gene mutations, the recommended therapy is tafamidis, and basically this is a small molecular therapy. So as you can see, there is an increasingly growing evidence that suggests the current HCM therapies rely on gene mutations. But as I pointed out, a significant proportion, approximately 50% cases are not known, and especially the Indian HCM genetic studies are underrepresented. So the focus of my lab is to identify new genes, key mechanisms, and therapeutic targets for hypertrophic cardiomyopathies and other cardiomyopathies. And the approach that we take is a patient-partnered clinical and basic science approach, wherein we collect a large number of family samples, and here in this case, trios, and isolate the DNA and perform whole exome sequencing. And we also do whole genome sequencing for the cases which are not answered by the whole exome sequencings. And once the gene is identified, we use the in vitro models, either stem cell models or other cardiomyocyte models, in order to understand the function of that particular gene variants. And once the gene variants function is established, we actually screen them in a larger cohort to establish their frequency and specificity. So if those variants are very common, more than 2% in a population, we generally create animal models, which include mouse, zebrafish, or fly models. So based on the focus, I had divided my team into genetics team and mechanistic team and therapeutic team, and these are the current team members of my lab. So unlike adult cardiomyopathies, childhood cardiomyopathies are life-threatening cardiac abnormalities, and they are very poorly studied group. And approximately 67% are with no known cause, and there is no therapy for these particular conditions. And among the various cardiomyopathy that affects the child, cardiomyopathies in Noonan syndromes are quite striking. So discovered by Jacqueline Noonan in 1985, the patients usually affected by this particular syndromes are having a short stature, webbed neck, and pulmonic stenosis. And many of these kids also have cardiomyopathies, and approximately 1 in 2,000 live births are affected with this condition. So in order to identify new genes for hypertrophic cardiomyopathy of such cases, we collected sample across India. First, we collected in South India, and we actually collected approximately 60 to 70 cases, but we picked only 20 cases in the discovery phase. The other 40 cases, we removed it based on the already known genes. And from the North Indian side, we have only 5 cases. For the discovery phase, we again collected 41 cases from Northern America from Lowry Lab, which is from UCSF, and we performed whole exome sequencing in these cases. And for replication phase, we actually collaborated with Rumiko's lab, and she actually contributed 35 cases. And from Europe, from Marco and Stefan's lab, which 160 cases, and from North America, from Bruce lab, 60 cases. And I want to point it out here that these case numbers are lower because we already removed already known gene mutations cases, which are not part of this particular study. So we took all these patients and along with the controls, and we performed whole exome sequencing. And here I had shown a diagrammatic representation of how we do the whole exome sequencing. We actually isolate the DNA, we sequence them, and we obtain the raw reads as FASTQ files, and we map them. And the variant calling is done by Picard tools. And Using GATK, we actually perform the variant calling, and ultimately we annotate them and filter them using the softwares which include dbSNP, 1000 Genome, or Exome sequencing project, or Korean sequencing project, Genome to MP, TopMed, and Nomad. So these are various human publicly available databases which comprise approximately 2,076,988 alleles. So what we did is we actually filtered those variants as shown here, and we ultimately got approximately 46 SNPs that are only observed in the patients compared to the controls. So we took the 46 variants of the disease, and it is given as Indian Database of Healthy Aging Nucleotide Sequence, idans.org. So we took this 46 SNPs that we obtained from the patients, and we filtered those into the idans.org, and then Genome Asia 1000K project and IndiGenome project. In this way, we obtained only 6 gene variants, and out of the 6 variants that are present only in the patients, we actually narrowed down to PRKCA. So parallelly, we also took already known genes and performed a network analysis. And for the network analysis, we actually took cluster 1 code and built by association. We combined them, we wrote our algorithm, and in that also PRKCA popped up as a candidate genes. So shown here are the various family members that are having the PRKCA mutations. The first mutation is serine 149 to asparagine. So the second mutation is E207 to G, and the third mutation is valine 566 to leucine. Basically, as you can see, the genotype-phenotypic correlation revealed that all these patients are young, and either they are familial or de novo in origin, and they all have hypertrophic cardiomyopathy, and some have mild rasopathy-like features, and others have a prominent feature such as short webbed neck and with valve defects also. So we could able to replicate that in the Japanese families also, as shown here. These are the 2 families, Japanese families, that possess the particular amino acid changes where arginine is converted to at the 260th position to cysteine, and valine from 566 to leucine. And this is the same mutation that we observed in the Indian family also. So genotype-phenotypic correlation reveal that both the patients are young, and both the patients are mutations are de novo in origin, and both the patients are short and have hypertrophy, webbed neck, and both have a combination of septal defects with hypertrophy, suggesting the complexity of the PRKCA mutation in causing cardiomyopathies and valve defects. So protein kinase C, actually PRKCA-C, codes for a protein called PKC alpha protein. So PKC alpha protein has 4 conserved domains, C1a, C1b, and C2, and kinase domain. And these C1a, C1b, and C2 are a calcium binding domain. As you can see, the mutations are widely spread, not concentrated on one particular region. However, the C2 domain has 2 mutations here. So the functions of this particular protein is majorly involved in metabolism, proliferation, apoptosis in heart, and they signals through RAF1 AKT ERK signaling, the RAS MAP kinase signaling. And they also phosphorylate troponin I, thereby they establish contact with the sarcomeric proteins. So we then took these mutations and we superimposed these mutations with the available structures. As you can see, the mutations are far away from the ATP binding domain, and this suggests indeed the whatever downstream effect that we are going to see, it's going to be allosteric in nature. So all these PKC mutations alter evolutionary concerned residues, and they're predicted to be pathological in nature. So far, people have reported PRKC mutants in the cancer. So in the cancer, these mutations are loss of function in nature. However, then we ask the question, what about the newly observed HCM mutations? So in order to achieve this, we actually collaborated with Shiv where we actually make the Fred sensor, and through the Fred sensor, here I had showing one example, E207G, where the Fred sensor suggests that indeed the mutated protein goes to the membrane and quickly comes back and induces the signals downstream. So this indeed suggests that they are gain of function effects, and through the kinase activity assay, also we confirm that it is basically a gain of function effect. So with Minhaj, we had achieved this particular protein as a purified form. Now we are actually waiting for the high resolution model for this particular protein. So as I said, the PRKCM actually activate the RAS MAP kinase pathways. So they phosphorylates RAF1 at a specific site, serine 499 region. And after phosphorylation, they heterodimerize. And once they heterodimerize, they again downstream phosphorylates ERK1 and 2. And parallelly, they can also phosphorylate AKT at the phosphorylation site 473 region. So we express this particular wild type PKC along with the mutant in the neonatal cardiomyocyte, and we perform the Western blot. And as shown here, the mutant shows increase in the phosphorylation of the AKT and ERK compared to the wild type. And this indeed suggests that the PRKCA mutants activate MAP kinase activity. So the PKC, as I said, the PKC alpha phosphorylates serine 499 region. So what we did is, so we actually make this particular phosphorylation site as a kind of non-active by introducing alanine and then co-expressing this along with the PKC wild type and the mutant. As shown in the representative blot, when you make this mutation alanine, so basically the phosphorylation of AKT and ERK actually are downregulated in the mutant compared to the wild type. And this data indeed suggests that the PRKCA mutants depend on RAF serine 499 region. So in order to know whether these mutants have a human relevancies, so we actually made a human cardiac stem cell models where these are actually made by Anupam. That's what I particularly want to show you this. So this particular, basically what we did, we actually took the ES or the IPS lines, we expressed those wild type and the mutant, and then differentiate those into the cardiomyocytes. And once the cardiomyocyte is achieved at day 8 to 14, what we actually basically do is like we actually measure the contractility and calcium signaling and the downstream signaling. So as shown here, the mutants actually shows increase in the cell size surface area compared to the wild type. And this is true for all the mutations as shown here compared to the wild type. And the mutants also increases the fetal gene hypertrophic markers. As you all know, when there is a hypertrophy, there will be a re-expression of the fetal genes in order to cope the pressure, especially the ANP and BNP in the human cardiomyocytes. As shown here, the ANP, most of the mutations activate the ANP and BNP compared to the wild type, and the mutants also activate the phosphorylation of AKT and ERK, and they also activate the phosphorylation of troponin I. So in order to know whether this happens in an in vivo model, we actually made a transgenic Drosophila model. As shown here, expressing the mutated protein, represented mutated protein in the fly ubiquitously actually shows a short stature compared to the wild type, and this is a reminiscent of the patients as shown here. And expressing this mutated protein in the fly eyes, we also see an increase in phenotype, and that is because of the increase in the R7 receptors. And these data indeed suggest these mutants are a gain of function mutations, unlike the cancer mutations. So we also express these mutations in the skeletal and heart muscles. As you can see, expressing the mutations in the skeletal muscles actually reduce the sarcomeric length in the mutants compared to the wild type. The length and the width is increased in the mutant compared to the wild type. Whereas expressing the mutated protein in the heart actually shows increase in the sarcomeric numbers compared to the wild type. So, and they also increase the heart dimension as shown here. So these data indeed suggest that the PRKCA mutations affect the skeletal and the heart muscles. So in order to know whether the signaling cascade is through only AKT or MEK, or is it through some other pathway also, to know that, we performed a rescue experiment where we actually express the mutated protein in the fly eyes and cross them with AKT, either AKT RNAi or MEK RNAi flies. As you can see, the AKT or MEK RNA flies actually shows a rescue pattern compared to the GMR expressing the mutant protein alones or the other representation mutations, V566L represent mutants also. So this indeed suggests that AKT and MEK pathways are the major downstream signaling cascade pathway that are activated in the PKC mutants, and thereby also confirms that indeed they are the gain of function mutations. So in order to develop a preclinical model to study the mechanism and the drugs, we actually made a transgenic mice that carries a representative mutation E2.G. As you can see, the transgenic mice shows increase in the heart size over here compared to the wild type. And we also measured their posterior wall thickness, as the posterior wall thickness is increased in the mutant compared to the wild type controls. And the mutants also, or the mutants recapitulate whatever we observed in the cell culture models, where the mutants show increase in the AKT and ERK compared to the wild type. And they also shows increase in the hypertrophic markers such as ANP and a shift in alpha MHC to beta MHC, which is expected in the hypertrophic per se, and also an increase in skeletal actin. So this indeed suggests that PRKC mutation induces hypertrophic cardiomyopathy through a gain of function effects. So then in order to know whether we can able to rescue this with a MEK RNAi inhibitor, we actually took these mice and treated the MEK RNA inhibitor 2 milligram per kg for 21 days. And this treatment actually indeed rescued the phenotype as shown using the ultrasound image here compared to the wild type. So the MEK treatment also reduces the increased EF fraction that we observed in the PKC alpha transgenic mice. And they also showed a reduction in the fractional shortening and compared to the wild type or transgenic PKC mice. So they also show reduction in the hypertrophic markers, which includes ANP and BNP, as shown in this picture. And in summary, these data suggest that PRKCA is a novel gene for hypertrophic in syndromic children, and Drosophila stem cell and mouse model recapitulate human Noonan syndrome associated hypertrophic cardiomyopathy phenotypes, and AKT and ERK pathways plays a crucial role, and MEK inhibitors shows efficient rescue of signaling, which is basically a preliminary data that we obtained so far. So now this can be used as a clinical diagnostic purpose. So I will stop here and I will take any question before moving to the second part of my talk. Any questions so far?
So, Dr. Dan,
Yeah.
if we look at some other questions in the meanwhile, I'll just ask two small queries.
Sure.
One is like, what is this MEK treatment?
So MEK inhibitor treatment is basically we had seen there is an increase in the MEK activation in these mutant flies and the mice and the IPS lines. So we actually took a MEK inhibitor, which is basically PD1035, basically an inhibitor that actually well proven. Now it's in the clinical trial. So we obtained that and we treated these mice, and it shows basically a rescue of the phenotype.
All right. Sorry for my ignorance. I just thought
no issues.
Right. And then you also mentioned about, you made a model where you try to, stop the phosphorylation of using S499,
Yes.
the RAF,
Yeah. Yeah.
and you said that it will rescue that
Yes.
again,
So
because it will that will prove that it's through RAF
yeah.
pathway, right?
Yes.
So what is this S499?
Yes. Yes. 499. Yes, basically, so okay, I got your question. So why don't I choose the RAF inhibitor rather than the
Right.
MEK,
Yeah.
right? So we didn't choose the RAF inhibitor because there is not a specific RAF inhibitor available so far.
Mm-hmm.
There is not I mean, specific to the RAF1, so RAF1 has three isoforms, RAF1, which is CRAF, ARAF, and BRAF. So there is no specific inhibitor for the RAF1, although there are few do exist, but still there is a controversy in that, and they don't work very well in the animal models. So that's why we went for a downstream signaling, which is a MEK basically, which is also because of many of the other pathway mutations in this particular family. For example, the RIT1 mutation or even the RAF1 mutation, people nowadays started using trematinib, which is a MEK inhibitor. It's a downstream of that. So we thought like, okay, that will be a strong thing to go ahead.
Yeah, I was just wondering, why did you choose
Yes.
S499 instead of just
Yes.
going to RAF?
So because of the non-specificity, and also RAF has multiple partners. So it's very difficult to target the RAF,
Okay.
so that's what.
Yeah, good.
Thank
So, what is the role of stem cells? You mentioned somewhere that embryonic stem cells.
Yes. So the role of stem cells here is, so these other things what we are showing is basically in the animal models, but it is not in the human relevant model. So that's what in a background of a human cells, whether this really happens or is the signaling are different basically, right? So in the background of the human relevance also, it is the same. Actually, it actually activates the ERK, and when you block it, it actually goes down, suggesting that this human model will help us to confirm that the pathways that are activated in the fly or the animal models are indeed an actual pathway that is existing in the humans also.
So for that, you made you had the embryonic stem cells, and then further worked with the some
Yeah.
organ.
So what I had shown is with the embryonic stem cells a model, but we also work with the induced pluripotent stem cell model. So we just recently got the IPS lines for one of the mutant, PRKCA mutant, which we characterizing now. It's almost like we have a achieved cardiomyocyte differentiation then, and now we are doing the Western blot on that, and they also show the same thing. And we also now have a CRISPR-Cas9 induced mutant model. So that will be another way to go. So that I didn't show much of the data here because it's all preliminary data, and all this data are unpublished. So that's what I really thought putting it in a very short way.
Okay, thank you.
Thank you.
So there is a question from my colleague, Dr. Gaurav Sharma. So he wants to ask, could you update us if the observed specific de novo mutants are a cause or consequence for the disease phenotype? That is that.
So okay, so it is basically a cause, because why I will say a cause. The cause is because when you compare, we had proven that as a cause in various models also, right? So we took first the cell lines and we asked the question compared to the wild type whether they can induce the hypertrophy. Yes, they did. And we went on to prove that in the transgenic fly model, right? If you introduce the mutated protein, clearly they introduce the hypertrophy. So, and to nail that, the mouse model also shows a clear hypertrophy. So it's a cause, right? So if it is a consequences, when you block the downstream signaling, like a MEK, which we are shown, so you will actually see still hypertrophy, right? So that's because it may activate through different pathway if it is a consequences. So that's why we conclude saying that this is a cause.
Okay, Dan, I think we can move to second part.
Okay. So what I will do, I will go to the second part. Okay, are you guys able to see the slides or no?
No, Dan.
Okay. Okay, now?
Make it full view.
So shall I start, Anupam?
Go ahead.
Okay, so what I had discussed so far is actually a new gene for the childhood cardiomyopathy. So now what I'm going to talk about is about another new gene that we discovered, which connects the hypertrophic cardiomyopathy with the diabetes. So as you all know that now, like it's a hypertrophic cardiomyopathy is a disease of heart muscle in the absence of the secondary cause. However, the recent reports, one which I showed here from the Michael Arrell group, suggest that there is an impact of diabetes on hypertrophic cardiomyopathy. And that's because 10% of hypertrophic cardiomyopathy cases are present or coexist along with diabetes. So we ask the fundamental question, is there anything or a shared genetic component for this? So in order to know whether there's a shared genetic component, we selected a cohort, which we termed here as cohort 1. And this cohort has a clinically diagnosed 300 HCM patients. And we actually measured the hemoglobin level in those cohorts. And we found out of 344 people have hypertrophic cardiomyopathy along with diabetes mellitus. And these are clinical data of this 44 is given here. And you can see that the glycosylated hemoglobin is average 9.2 in these patients. And this approximate to 15%, which is slightly higher than what my Dr. Michael Arrell group had shown, that indeed 10 to 15% now of HCMs are associated with diabetes. So what we did, we took the 44 patients and along with the 80 available respective healthy controls, and we isolated the DNA and performed exome sequencing. So to the same kind of filters that I shown earlier, we actually narrowed down to one particular gene called adiponectin receptor R1. So as shown here, this particular patient P1 possessed a particular variant where this particular nucleic acid change converts the amino acid from leucine 157 to histidine, which is a highly conserved amino acid. So the patient who carries this also has diabetes, and we actually got his family members. As you can see, one of the family members, along with the P1, they also have hypertension, but the other members are negative for any disease. However, the mutation was only present in the HCM with diabetic patient P1. So then we want to know whether the same gene is implicated in the other cohort, an independent cohort. So we selected cohort 2, where this cohort has HCM with diabetes, which is approximately this time 99 in number. And we found 2 patients in this particular cohort also carries adiponectin receptor R1 mutations, where adiponectin receptor R1 at the position val in 164 is converted to methionine. So next we took cohort 3, where we actually asked these questions. So what about HCM alone cases? So in this HCM alone cases, which is 380 in number, we found another mutation that has another variant, which I showed here, phenylalanine 145 region 2 isoleucine. So we then asked the question, what about a cohort with diabetics without hypertrophic cardiomyopathy? So in this cohort, there is approximately 800 in number. So these particular variants are not present in diabetics without hypertrophic cardiomyopathy. Whereas cohort 5 and 6, which are a secondary hypertrophic cardiomyopathy and dilated cardiomyopathy, we didn't observe this particular variants. And this indeed suggests adiponectin receptor variants are associated highly with hypertrophic cardiomyopathy with diabetes compared to HCM without diabetes. So then we actually look into the allele frequency of this particular adiponectin variants across the globe using various population genomic database such as 1000 Genome, NHLBI, COVA, EXAC, and NOMAD, TopMed, Genome to MP. And for an Indian specific, we actually looked into Genome Asia and our own data set, IDANS and South Asian Healthy Controls. So as you can see, the L157 histidine and the F145I both are novel, whereas the V146M is basically present in a ultra rare frequency in the NOMAD and TopMed and Genome MP. So then what we did is we actually performed an odds ratio comparing the primary HCM with and without, comparing with the other population genome data. As you can see, the odds ratio is 9.7 in the primary HCM alone cases for the presence of this particular variance in that particular cohorts, suggesting that indeed, if the patient has an adipose receptor R1, he will be increased risk factor for hypertrophic cardiomyopathy with diabetes. So shown here is the family members carrying the particular variants. As you can see, the P1 family, it is de novo in origin, whereas the P2 and P3 family, the particular variant is actually inherited, whereas the P4 it is not. And P5, which is actually an elderly lady who's actually affected with hypertrophic cardiomyopathy alone without diabetics. So, but their family also, it is de novo in origin. So as you can see, all these particular variants are evolutionarily conserved, and they actually affect the protein structure and function through various software, which I had mentioned here, which includes SIFT, Polyfen2, Panther, Mutation Tester, Metadome, PMUT, and CAD. And this indeed suggests that the adiponectin variants indeed are associated with hypertrophic cardiomyopathy with diabetes and without diabetics also. So what exactly adiponectin receptor R1 is doing in the heart? Adiponectin receptor R1 plays an important role in the heart. It is basically a 7-membrane protein, like as I shown here, which is equivalent to that of the GPCR protein, but in an opposite topology. They're actually their agonistic is adiponectin, and through the adiponectin, they actually activate the downstream pathway signaling cascade, which include AMPK pathways, which include AMPK and acetyl-CoCarboxylase. And parallelly, they can activate the insulin pathways, which are IRS and AKT GLUT4 and phosphofructokinase, and thereby ultimately controlling the glucose and fatty acid metabolism. So in order to know whether this mutation can induce cardiac hypertrophy independently, so we actually construct the virus and we express these viruses in the wild type and mutant virus in the primary cardiomyocytes. As you can see, expression of these mutations increase in cell size compared to the wild type and beta-gal expressing cells, and they also increase the hypertrophic markers, which include ANF, BNP, and beta-myosin heavy chain. And this data indeed suggests that the adiponectin variant can also cause cardiac hypertrophy. So next, in order to know the pathways that are activated for adiponectin receptor variants, we perform Western blot in the downstream signaling. First, we actually check the basically, since they activate the AMPK, we first check the AMPK. Surprisingly, these particular mutants, they don't activate AMPK. Rather, they choose another pathway, which is P38 mTAR pathway. As shown here, they activate the P38 mTAR pathway, and that is only for the hypertrophic cardiomyopathy with the diabetes. In contrast, for the HCM alone mutants, which is 145I, we didn't observe AMPK or P38 or mTAR pathway activation. Rather, it actually chooses another different pathway, which is ERK pathway. So these data indeed suggest the adiponectin variant activate diverse pathway. If they activate P38 mTAR pathway, they actually go into hypertrophic cardiomyopathy with the diabetes. In contrast, if they don't activate these pathway, but if they activate ERK pathway, then that particular patient only have hypertrophic cardiomyopathy. this indeed suggests the position and the location of the mutation is very specifically important in these receptor mutants. Next, in order to know whether the adiponectin receptor R1 diabetics association mutant affects the glucose metabolism, we actually measure the 2D uptake, which is a glucose uptake in the cells in the mutants compared to the wild type and beta gal expressing cells. As shown here, the mutants actually are not able to uptake it properly compared to the wild type, suggesting they are compromised compared to the wild type with or without treatment with adiponectin. And also, we measure the downstream signaling cascade, which include insulin receptor substrate 1, and especially serine position here. So the serine position is a determinant factor for the insulin sensitivity here. So you can see there is an insulin sensitivity here for the particular mutant compared to wild type and beta gal expressing cells, which is also reciprocated in the downstream deactivation of the phosphorylation of AKT and phosphofructokinase. So this data indeed suggests that the adiponectin receptor R1 mutation affects the glucose metabolism. So in order to know that whether this happens in a in vivo setup, we actually made a Cree mice. So the Cree mice actually showed where they're normal when they are born, and when we treated with tamoxifen at a particular range, the Cree mice actually shows a kind of hypertrophic accompanied by dilatation. So then, since this particular mice also shows a dilatation phenotype accompanied by dilatation phenotype, we suspect that the hypertrophic cardiomyopathy patients with the diabetics will be propensate towards the dilated cardiomyopathies or heart failure. So as you can see, the fractional shortening in the mutants are actually basically is increased, and also the cross-sectional area where you can see in the mutants are actually increased. So since the mutant activates the downstream mTOR pathway, we treat them with rapamycin. So the treatment of rapamycin actually rescue the phenotype as shown in the ultrasound image, where you can see a decrease of posterior wall thickness, and also the fractional shortening get normalized, and the cross-sectional area also got actually decreased from the increase in cell size. So we then check the markers, basically the calcium signaling proteins, which include phospholamban and SARCA2 in this case. As you all know, the phospholamban and SARCA are the two antagonistic protein, meaning like if the SARCA is increased and the phospholamban is reduced, they actually involved in regular calcium signaling, whereas if it is opposite, the calcium signaling is actually affected. As you can see, the SARCA is basically increased in the mutants, whereas it is decreased in the treated animals, basically. So we also see an increase in hypertrophic markers, which is ANF and vitamin C, which are rescued by the rapamycin. So since fibrosis is the main phenomena in the hypertrophic per se, we actually check the mutant for fibrosis. As you can see, there is a rich fibrosis in the HND staining, and this fibrosis are rescued with rapamycin. And for collagen area fraction, this is basically they also reduced. So they also reduce the mTOR signaling, where I had shown here with the Cree vehicle and rapamycin. The rapamycin rescued the phenotype, and then so this data indeed suggests the rapamycin rescued cardiomyopathy phenotype in adiponectin R1 mice. So this data indeed suggests that the adiponectin receptor variant causes hypertrophic cardiomyopathy, and it depends upon the mutation and the location. And if the mutations are activating P38 mTOR, then they actually causes hypertrophic cardiomyopathy with diabetes. And if the mutations are activating ERK, then they only causes hypertrophic cardiomyopathy. So this indeed suggests that adiponectin receptor R1 plays an important role in hypertrophic cardiomyopathy with the diabetes. Okay, I'll stop here, and thank you very much. So I will take any questions.
So then it was wonderful. I still have a couple of queries. If Anupam can allow me ask directly.
Yes, sir. Sure, sure.
So Dan, is it a model like for type 1 diabetes or the other one, type 2?
So I would think that it will be a model for type 2 diabetes.
Why do you think so?
Because there you see an insulin resistance there.
Why I ask this question is that type 1 diabetes is more genetic, right? Type 2 is not.
Yeah, so one would expect that type 1 is genetic, but if you see the age of these patients, like they are actually above 60 and one is above 42, 45, something like that. So especially the patient in the third family. So that's what I suspect that it might be a type 2.
Yeah, although they are quite related. I mean, the people who have developed type 2 diabetes,
Yes,
basically run into families, but they carry the risk of having,
yes.
excuse me.
Yeah. So as you pointed out, it is a very fine line to differentiate these two, and I would agree that. So the thing is like, it might be useful for some type of diabetics, but what I would see that is like what I observed in the patients with the HCM patients, that will be a good model to actually exemplify here.
Okay, so then I have just a minor query about your cohorts. You have cohort 2 and cohort 3 where you have the cardiomyopathy with diabetes and without diabetes. So you said that the frequency of this mutation, which you found the ADPOR1 V164M in only 2 of the 99, subjects you
Yes.
there. So,
Yes.
and the other cohort is still lesser, 1 in only 380. So, with that low frequency, how would you what would you like to say? How would you, like to defend
So,
hypothesis?
yeah, it's interesting because this is a chart that I had shown for the frequency, because as you can see in Nomad, right? It's a general population across the globe that they took. Out of 2,44,522, their frequency is just like 0.0001, meaning like one in it's ultra rare basically, right? Whereas in the Indian population so far, whatever we screened, you can actually see here 1,196 or 188 or 3,600. This particular mutation is not observed in the Indian patients. And I will caution you here, the frequency here, for example, Nomad or TopMed. TopMed is a disease database. They compile a lot of diseases there. And Genome2MP is also a disease database. Nomad also has a lot of cardiac disease people. So it's a random disease people. So I would say that the frequency is ultra rare and it's completely absent in Indians. But when you make an odds ratio, the odds ratio is also very high. So it increases the risk of having that particular condition.
Mm-hmm.
So in that way, I think it's statistically very significant in causing the disease.
Oh, okay. Thank you. Yeah, wonderful talk.
Thank you very much.
So there are no more questions in the chat or Q&A. Any faculty want to ask, they can directly ask the questions. There are people from cardiology, CTVS, I can see. Okay, Dan, I think there is no more question. I would like to thank you for accepting this invitation.
Dr. Prashant Sharma is asking some questions.
Okay, sure.
Hi, sorry to interrupt you, Gaurav. Thanks, Dan. That was really interesting. I have a question which may be a little out of your area of expert, the area that you talked about today. But we have these patients with clonal hematopoiesis, and they have acquired mutations in their hematopoietic stem cells, and they have an unusually high frequency of cardiovascular and cerebrovascular thrombotic events. And there's no data from India yet on the frequencies or the effects of these acquired mutations, the somatic mutations. Any thoughts on that?
Okay, that's a wonderful, wonderful question, and that's an area that we are working now. So that's we basically are talking about the CHIP genes, like a cluster of clonal hematopoietic gene that actually causes a coronary artery disease. We actually had done in collaboration with MIT. So we actually had done thousands of exomes with coronary artery disease, especially premature coronary artery disease. So we do observe the CHIP genes involvement in the Indian patients too. And the frequency is very interesting because we just learned in another project, another graduate student is working on that, Binay, basically. So we just learned that the frequency of, there are many CHIP genes. So basically the ASXL2 is basically, people had shown that it's highly involved, like in the Western population, and TIT are the major genes. But here it seems like there is a shift in this percentage that we are observing. And we are also observed a new player in that, so that will be another day for a wonderful talk with you. So yes, so we do have data on that. So the answer to your question is, especially for the coronary artery disease, unlike the Westerners, so the CHIP genes are associated, but it is associated in a different percentage. What percentage, what they are showing is very different here. Especially there is a complete flip between the TET2 and ASLX2.
Thank you.
So any more questions, please? Okay, then I would like to give you a vote of thanks on behalf of our department for accepting our invitation.
So one thing I forgot to tell, and most of the work is, a lot of these works are established by Anupam when he's over here. So, and I think like you guys had picked the right candidate for the department, and I'm very proud of that. So thank you, Anupam. I think you will do a wonderful job.
Yeah, thank you, Dan. And I'll thank you, and I'll thank my department head who gave us chance to start this series, and my colleagues who are supporting me regularly, Dr. Aruna Rakha and Dr. Gaurav Sharma, who are also here, and our departmental student. Thank you.
Thank you, sir. I wish one day I will come over there to meet Professor Sunil there.
We would love to have you with us.
Yeah.
So unfortunately, we cannot at the moment, but it will be a wonderful idea if you can come over and we meet in person. So thank you very much for accepting and giving this wonderful talk. I'm sure many would have really enjoyed this. I personally enjoyed each part of it. So that's why I was listening very keenly and going through your slides. So pick up some few questions as well.
Yes.
So very nice.
You.
Although I am not a person of genetics, so I am an immunologist, so I don't understand much part of it. But of course, your explanations were very nice, and I could understand quite a bit of it. So that means maybe others might have understood much better than me. So thanks.
Thank you very much.
Anupam, and thank you for endorsing Anupam. So I also found Anupam a very hardworking guy. So let's see how he goes about. Thanks for his training in your lab.
Thank you.